Hi, my name's Gunn and I work in the Laboratory of Molecular Biophysics at Uppsala University. May I start by just saying how interesting I think you are? And I find you interesting because I know that every single carbon atom in your body has been transformed by an enzyme called Rubisco, and I'm really quite interested in Rubisco. And that's because Rubisco performs the initial carbon fixation step of photosynthesis, converting carbon dioxide into energy in the form of carbohydrates. But there's a problem with photosynthesis, and that is that oxygen is produced as a byproduct, which is great for you and me because it means we can breathe, but it's not so great for my friend Rubisco. And that's because this is what carbon dioxide and oxygen look like to a Rubisco. Rubisco has a really hard time trying to tell them apart, and this makes it really inefficient. In fact, Rubisco often limits the growth rate of plants. Now, once photosynthesis arose, this led to a rapid increase in the amount of oxygen in the atmosphere, and this competing oxygen reaction became problematic. And this led to a diverse range of carbon-fixing machines that evolved in different organisms to face the challenge of all this pesky oxygen. And here are some examples of some different carbon-fixing machines that have evolved in different organisms, and you can see that they come in all sorts of shapes and all sorts of sizes. What I'm interested in is to understand the process through which these Rubiscos adapted to a changing environment and how we can use this information to build a better Rubisco. Which Rubiscos are really good, which Rubiscos are maybe not so good, and what aspects can we borrow from different Rubiscos, from different lineages, to try and build a better Rubisco. To look at Rubisco structure, I use protein crystallography, where I force my Rubiscos to grow as crystals. And these crystals are just lots of copies of Rubisco proteins packed in a really regular arrangement. I can then hit these crystals with X-rays, and the X-rays bounce off each atom in the crystal at different angles. And it forms these pictures that you see here in the top right, called diffraction patterns. So each spot in these diffraction patterns gives us information about the location of each atom in the crystal. I can then use a mathematical lens to focus these diffraction patterns and reconstruct what the protein looks like. And here's one of the Rubiscos I've been working on recently. And what I found here is that this particular Rubisco has a completely unique locking mechanism, shown on the bottom right. And this Rubisco's got 10 of these locks that hold them together, and we think it makes it more stable. What I'm trying to do now is to introduce this protein glue into other proteins to lock them together and make them more stable. So Rubisco's function is so limited, and yet it's so fundamental for life as we know it on this planet because it converts carbon into a form that our bodies can use. By studying the evolution of nature's carbon-fixing machinery, I'm trying to build a stronger, faster, fitter Rubisco to improve the efficiency of crop species, the efficiency of biofuel-producing algae, and for use in artificial photosynthesis systems. So thanks for listening, and we'll see you next time.